For this session, we're going to take an initial look at our blood vessels in circulation. So to get started, let's take a look at the overall systems and vessels. All right, so for our blood vessels, these are going to help us conduct blood between our heart as well as the peripheral tissues. And we have three different blood vessels that we'll take a look at. Those are our arteries, which carry blood away from the heart, and we can also term these as efferent vessels. So when we think back to our neural chapter and looking at our neurons, we had afferent and efferent neurons, and we name them the same way. So these arteries can be efferent vessels because they are actually carrying the blood away towards the effector cells. Okay, so those cells are what are waiting to receive these nutrients as well as our gases, so oxygen. All right, so we're moving that blood out and away. And then on the other side, we have our veins, and the veins carry blood back to the heart. This is our deoxygenated blood. Okay, and so now we can see these as our afferent vessels. All right, and in that case, that afferent is moving back towards the heart, kind of seeing the heart as the control center. All right, so like I said, afferent vessels go with veins. And then we also have our capillaries. All right, and the unique thing about the capillaries is that this is the only place that we can exchange substances between our blood and tissues. All right, these are our smallest vessels that we have. They interconnect the smallest arterioles to the smallest venules, and we get that exchange in this area. All right. Beyond this, we have our two circuits. The very first one was our pulmonary circuit. And on our pulmonary circuit, this is where we have our deoxygenated blood coming out of the right side of the heart, moving into this pulmonary circuit in order for us to exchange gas with the lungs. All right, so moving from a deoxygenated state of blood into an oxygenated state of blood and returning it now back towards the left side of the heart. And then we also have our systemic circuit. So moving from the left side of the heart, coming in from those pulmonary veins with our oxygenated blood, getting pushed out by our left ventricle into the aorta and moving out from there, moving this blood that now has oxygen abundance to it towards our cells. All right, and once we have exchange at the capillaries, we're also going to pick up the carbon dioxide and move that through the veins back towards the right side of the heart. Okay, so that's what we look at for our pathways. All right, moving on from that, now we can actually take a look at these arteries and veins. All right, and there are different layers to our arteries and veins. And on this image right now, the artery is represented in red and the vein being represented in blue. As I go through the different layers of these arteries and veins, I'm going to highlight them so you'll see that they'll pop up blue, and then you can also keep an eye on the right-hand corner, and that'll actually tell you the name of the item that we're looking at. All right, so this very first one, since I'm on the innermost layer, and this is consistent between the arteries as well as the veins for the innermost layer, and you're going to see a lot of it's consistent, but we'll talk about differences as well. Okay, but this is our endothelium. Right, so we see it here on the artery, as well as if I come over to the vein, there's the endothelium. All right, and that endothelium is consistent back through to the heart. All right, so we still also have that endothelium on the inside of the heart as far as its innermost wall, and so it's this continuous track of endothelial cells. All right, and within this endothelium, we call this our tunica intima or tunica interna. Okay? And the endothelial cells also have connective tissue in there that has elastic fibers because we're going to want some expansion and distortion in here. All right, and we find a lot more of these elastic fibers within our arterial side. Okay? So if I actually look at this, as I said, we're still within this tunica intima, so on that tunica intima, in our big arteries, we can also find this subendothelial layer as we move down, so it's a fairly thick layer. Over on the vein side, we really don't see that. Okay, and then on this arterial side, zoom in a bit, because we actually have this thin layer in here. That's our elastic membrane that exists right there, but this is seen as our internal elastic membrane as part of that tunica area. So it kind of borders between the tunica intima as well as our tunica media. All right, so we really see it as truly part of that tunica intima. But then we can move on to the next layer, being the tunica media. All right, and this is seen as our middle layer. So we have it here, all right, and over on the vein side, we can also have it here. And like I said, on the large veins, we also have that elastic fiber right there, right? But onto the tunica media. So for this tunica media, right, this is our middle layer. This is where we find our smooth muscle. Okay? And this is where we can have influence from our sympathetic and parasympathetic systems, because when we actually have contraction or vasoconstriction, this is going to decrease our vessel diameter. And that's all happening here at this tunica media. Or we could have relaxation, which then the vessel diameter increases, or we term that as vasodilation. All right. But then as we move from this tunica media, as shown on both of these, we'll find another layer of elastic tissue. All right. And this is now going to be our external elastic membrane. All right, so that's a divider between our tunica media to the tunica externa. Right, and we need to have these elastic membranes in there. Even though we see our arteries as kind of our stiff vessels, right, because they're going to be under a lot of pressure versus our veins are actually under lower pressure, and these can deform quite a bit. Right, these arteries kind of in a cross-section hold their round shape, whereas veins are going to be distorted, not being under so much pressure, they can be manipulated. But these arteries holding this round shape to them, these elastic membranes allow for when that heart pumps and basically pressurizes the system, they can have this distortion, right? They can deform. They can actually expand and contract as needed as the pressure increases, that we're not actually pressurizing the entire vessel with this fear that it's going to burst if we hold this constant round shape. So that's why we need those elastic membranes. All right, now moving on to that external membrane, or the tunica externa, or our tunica adventitia. All right, this is that outermost layer. This is connective tissue. All right, within our arteries, it mainly contains collagen and scattered elastic fibers, and that collagen is what really helps it to hold that shape. Okay? In our veins, we actually find a thicker layer, all right, relative to our tunica media. All right, but in those veins, it's a bit thicker. Okay, within the veins, this tunica externa contains networks of elastic fibers and bundles of smooth muscle cells. All right, so right there, collagen in our arteries, elastic fibers and smooth muscle cells on the veins. That tells you that there's much more distensibility in the veins. Okay, but also on this outside, that is where we can see it anchoring to our surrounding tissues. Okay, and one thing that's not really noted on here as we get into our large vessel walls. Okay, that's you can see it looks pretty smooth and clean, but actually on our large vessel walls, as we get towards this outer area, what we may find is what we term as our vasa vasorum. All right, and that vasa vasorum means vessels of vessels. And so we would actually find small arteries and veins on the outside of these large vessel walls, because since we have this epithelium that is continuous through the inside of these arteries and veins, they cannot nourish themselves. All right, that our blood will not pass nutrients through there. So there would actually be arteries and veins lining the outside, or that tunica externa, then providing nourishment that can be brought inward to make sure that these cells receive oxygen as well as nutrients. All right, but that being the vasa vasorum. All right, a couple other things to talk about kind of from this point of view is how we actually move down. All right, so if I'm starting in this area right here that we're already looking at, this cutaway area, we can see that as kind of our large artery. So when you think about the aorta, right, so that aorta comes out of the left side of the heart, but then it goes up and around its arch, but then continues inferiorly. All right, as we're in that area around the arch and as it starts to drop down inferiorly through the trunk, we do find this to be an elastic artery. All right, these are large vessels that are close to the heart. All right, and these ones have that ability to stretch and recoil when the heart beats. Okay, so we'd see that there, basically within the aorta, but we also see it within our pulmonary trunk. Okay, that pulmonary trunk is also an artery, and it's going to be highly pressurized with that blood coming from the right ventricle. Okay, and then we also see it within the branches that we find close to those areas. So right off the pulmonary trunk as well as right off the aorta. But then as we move down, right, so those elastic arteries being our large vessels, our muscular arteries, so if I rotate this view, you're going to see that this gets smaller and smaller. Okay, so it's still being my artery as it's moving down, but as we move down through the body, all right, as that aorta continues to descend downward, it's going to get a little bit smaller. So we'd see it as a medium-sized artery, and this is now our muscular arteries. All right, and these muscular arteries, we start to lose some of these elastic membranes. Okay, so right here where we talked about those elastic membranes inside. All right, and these muscular arteries distribute our blood to the skeletal muscles and internal organs. All right, and then moving on, as we get away from our arteries, we then start to get to smaller types of arteries. Right, so these are all arteries, but as it gets smaller, then we get into an arteriole, and the arteriole is down towards the end area of this arterial pathway. For our arterioles, they have poorly defined tunica externa. All right, reason being is that they're shifting from these arteries that are highly pressurized. Pressure is going to decrease. As we move into that arteriole, we lose that tunica externa, so it's really just this smooth muscle that we see on the outside of it. Okay, so these arterioles, and then our tunica media is only about 1 to 2 smooth muscle cells thick. All right, so it's very thin layering. Reason being is because after we leave this arteriole, then we shift into our capillaries. All right, and the capillaries being our smallest blood vessels, and these are the only blood vessels to allow exchange between our blood and interstitial fluid. And they have very thin walls. We're going to talk more about them as we look at the differences in capillaries and also how we pass our nutrients as well as gases through them. All right, but we make it through the capillaries, and then we come back to the other side where we'll run into venules. All right, these venules are small veins. For these venules, the veins are smaller than about 50 micrometers, and they lack their tunica media. All right, and what they really look like are kind of expanded capillaries. But these venules are now collecting blood back from the capillaries. All right, and once we make our way through this capillary bed right here, you can see that the coloring, let me click off it, coloring shifts from red to blue, because that's where we start to lose our oxygen, pick up carbon dioxide, and we now have our deoxygenated blood coming up through these venules and then up into our veins. Okay, and we can also see a division on the veins like we saw for our arteries, that we have medium size up to large size. All right, so moving our way back up, we look at our medium sized veins. All right, and these ones will now start to enlarge from those initial venules. These range from about 2 to 9 millimeters within their internal diameter. These have a thin tunica media. All right, and with that tunica media, it's got both the smooth muscle cells and collagen fibers. And the thickest layer here is the tunica externa. All right, and within that tunica externa, we now see longitudinal collagen and elastic fibers. Okay, rather than laying in kind of a cross view. All right, but then moving back up and around, we would then get back to our larger area for these veins. So our large veins, they once again have all 3 vessel wall layers, seeing that tunica intima as well as the tunica media and tunica externa. Okay, it's got this thin tunica media surrounded by a thick tunica externa. And areas where we find large veins would be our superior and inferior vena cava, as well as the tributaries that connect right into those. All right, and that's what we see for our arteries, moving to arterioles, moving to capillaries, back up to our venules and into our veins. So that is the pathway that we take. All right, out of the heart from the left side and back around, coming back into the right side. All right, so now let's talk a little bit more about capillaries. So for these capillaries, all right, if I want to take a kind of regular view of a capillary, for our typical capillary, it's going to consist of this tube of endothelial cells that have a very delicate basement membrane. So if I'm looking at this right here, here's my basement membrane. All right, really thin. You can see it's only 1 cell thick as far as that basement membrane, and then on the endothelial cells, we see the same thing, just a single cell. All right, average diameter on these is about 8 micrometers, okay? And that's about the same diameter of a red blood cell. All right, when we talked back about our blood and our red blood cells going through, we talked about rouleau, where those red blood cells get stacked. Well, that's how they come through our capillaries, kind of in this stacked fashion. All right, and we have two different types. We have our continuous capillaries, and then we also have our fenestrated capillaries. And then we'll also talk about sinusoids. All right, but for our continuous capillaries, starting over here, all right, the endothelium itself in here is a complete lining. So you can see it on this cross view right here. And we find these throughout the body in all of our tissues, with the exception of epithelia and cartilage. Remember, cartilage is avascular. Same with our standard epithelia. Okay. But with this continuous capillary, it's going to permit diffusion of water as well as small solutes and lipid-soluble materials. Okay. Continuous capillaries prevent our loss of blood cells, right, as well as plasma proteins. There will be selective vesicular transport through our capillary, or these continuous capillaries. Okay. And we also find very specialized continuous capillaries within our central nervous system, as well as in the thymus. Okay. For these, they have tight junctions within the endothelia, and that really helps us to enable restricted and regulated permeability. So that's our continuous. Then we have our fenestrated capillaries, and that term fenestra from Latin means window. So with these ones, what you can see is there are these pores that exist. So that endothelial layer is not continuous like we see back on that continuous capillary. All right, and with this fenestrated capillary, it's going to allow for a rapid exchange of water in larger solutes. Okay, and we find these in different areas. All right, so these are very specialized areas. So within the choroid plexus of the brain, as well as within our capillaries that interact with the hypothalamus and pituitary glands, as well as pineal and thyroid glands. Okay, so that way we can actually pick up those larger hormone molecules. And then we also find our fenestrated capillaries within our absorptive areas of the intestinal tract, so down in the small intestines and large intestines. And then these are also located within our kidney filtration sites. So those are fenestrated capillaries. And then we have our sinusoids. Okay, these sinusoidal capillaries, right, they are very similar to our fenestrated capillaries over here, all right, except for these sinusoids are flattened and irregularly shaped. All right, and we'll also see these gaps that exist in here, all right, where we did not see that on the fenestrated capillaries. All right, and also for sinusoids, we may or may not have a basement membrane. If it is there, it's very thin. These help to allow more water in solute exchange. So those plasma proteins can actually get through these sinusoids. And where we mainly find these is within the liver, but we'll also find these in our bone marrow, in the spleen, and also other endocrine organs. All right, once again, allowing for these larger hormones to pass through, keeping in mind that those hormones could either be amino acid derivatives, they could also be polypeptides and steroids. So we start looking at much larger molecules. All right, so that's kind of the overview of a capillary, but now let's look at how these capillaries interact with our arteries and veins. Okay, so what we have are capillary beds. All right, and so this is kind of that cartoonish drawing, but over here, this is actually under a light microscope where we can see our capillary beds that exist in here. And so we can identify this artery as well as an arteriole, and then the capillaries that branch off from there. Okay, for our capillary bed, it's this interconnected network of capillaries. Okay, so it's not that we just have this single artery coming down, then run into an arteriole, and then have one pathway through a capillary to get back over to our venule and up to the vein. Right? It's this interconnection. It's kind of like a spider web connecting through. And one thing it does is it makes sure that we have an exchange surface between these cells that these capillaries interact with. So if we only had one going across, then you'd see that there'd be a lot of cells that the capillaries are not touching. And also keep in mind that this right here is a two-dimensional view. So this is just a slice that if we were to look at it in depth, that we'd see more and more capillaries the deeper we go, and could be more interaction between these cells at different levels. All right. And keep in mind that a capillary bedding, though this image only shows a single artery coming down to these arterioles, that these beds may be supplied by more than one artery. Okay? And when we see multiple arteries, we'd see that as collaterals. Okay? So here we have two arteries coming into a junction, then into the arteriole, So that's a collateral, where they start to connect with one another. And so they fuse before giving rise to that arteriole itself. All right, and the other term that we'll use is as they fuse right here, that's an anastomosis. All right, so anastomosis is when two vessels come together, all right, or the joining of blood vessels. All right, but in having multiple arteries coming down and having an anastomosis, this allows for a continuous delivery of blood to the capillary bed, even if we have blockage within one artery, or if that artery may be compressed for some reason. Okay, think about it that, and then we may all run into this, but, as you're maybe sitting there on the toilet for too long of a period or sitting at a desk for too long of a period, and you start to lose sensation in your feet, all right? You have not lost total blood flow. If you look down, your feet should not be blue at that point. They should not be cyanotic, but you may be losing blood flow to certain areas. Just a matter of, in having that compression on the arteries, there's other pathways that the blood can take to feed other cells. Okay, so we don't always continuously cut it off because of these other pathways and having these collaterals. All right, but as we come down and we look at this capillary bed, one other thing we can note is that we have this arteriovenous anastomosis. In other words, that is a bypass from the capillary bed, that we would have blood that flows straight across and goes back up. All right, so we can always have these bypasses. All right, and that can be regulated by sympathetic innervation. And back to sympathetic, remember that's our fight or flight. That with that, we want to make sure that we have a lot of blood flowing. Well, when we start to think about our blood flow in the sense of where we need it to go, what can happen is we could have vasoconstriction along these capillary beds, right? And then vasodilation to this arteriovenous anastomosis, and we now kind of just have this blood shunted up and away and back on its way, returning, all right? So that way we can, and it may be returning to some other area. So it's just a matter of how can we actually shift that blood flow around, right? But that's all under sympathetic innervation. All right, and then we also have what's termed as our thoroughfare channel, all right? And that is the most direct passage through the capillary bed, whereas you see all these other ones branch out. Okay, the thoroughfare is that kind of direct passage. All right. Another item to talk about is our medarterial or precapillary arteriole. All right, and that would be this area right here. All right, so that's our precapillary arteriole. So being the initial segment of our connection passageway to allow blood to flow in. All right, this area still contains smooth muscle that can change the vessel's diameter and adjust our flow rate. All right, and connecting that precapillary arteriole to the capillaries, we find precapillary sphincters. All right, so these are bands of smooth muscle that contract and relax to control the flow into the capillary bed. And once again, like I said, as far as shutting off that flow in order to shunt that blood. All right, how do we need to move it? Or maybe we need it to move further down this passageway. All right, we can actually constrict all of this to get that shunting. All right, that's what we find for those precapillary sphincters. All right, and then what we also have in here is what's termed as vasomotion. All right, and that is cycles of contraction and relaxation that kind of helps to pump the blood through. All right, and you can see these precapillary sphincters throughout. All right, and that helps with that vasomotion, that we get this pumping action. All right, another thing to note while we're looking at this slide is that, a difference between being sedentary and highly active. One thing to note throughout your entire body, all right, right now, your body has all the capillary beds that it's going to have. If you have damage, then yes, we can have some genesis going on as far as reformation of blood vessels because they can retunnel through when we deal with damaged tissue. But for the most part, all right, so let's say you're sedentary right now. What you may notice is that these precapillary sphincters are going to have certain channels cut off. So it may actually have constriction here continuously to block off this passageway that, running through the thoroughfare is going to be enough blood and interface to deliver oxygen into the extracellular or interstitial area that can then interface with all these cells. And those cells are okay. They're not under high demand. They're not going through a lot of cellular respiration. So that's okay. But as soon as you start to exercise and become more active, well, these cells may need more oxygen. And that's where these precapillary sphincters would then open up and start to open up these channels that we're now delivering more and more oxygen. It's going to flow through at a fast rate, and not just the oxygen, but also the nutrients. Okay, so that's another item to keep in mind as far as how these capillary sphincters work. All right, moving on from our capillaries, we can now take a quick look at the venous system. All right, and the thing to note about the venous system is that our blood pressure is reduced after it moves from the arteries, where it's under a high pressure, into the arterioles, pressure is reducing. When it gets to the capillary beds, it's fairly low. And then as it moves over to the venous side, it's really low. It's only about 10% of what it was coming out of the ascending aorta to when it arrives at the venules. All right, so now it's down to about 10% of that. So very low blood pressure on the venous side, or venous side. Okay, so now we need some mechanisms in order to get that blood flow back to the heart, and especially in the lower limbs where gravity is pulling on this blood. All right, so one of those items would be veins. All right, so I'm sorry, not veins, will be valves. Okay, so we have these valves right here. All right, and those valves are folds that we find within that tunica intima. All right, so they project in from the vessel walls. And so I can come back and show you that on here. So here were those valves. All right, so you can see it, and this is actually looking down. If we look at it kind of from the other end on that valve, what you would see is that that valve is shaped just like cusps of the heart, okay, that these are going to work when we have backflow. So if we actually have blood flowing up towards the left corner of the screen right now, as soon as we depressurize on each heart stroke, as that blood would start to fall back down from gravity, these valves would close. All right, so we can come back to this point of view, that once again, if I have blood that we are trying to pump upward, okay, as we are in diastole and blood pressure is reduced, all right, as this blood flows back down, that's poor color to choose, all right, so as it flows back down, that's when the valves would shut, all right, but then we'd actually be able to hold it in there. Okay, these valves help to ensure one-way flow of the blood back towards the heart. And then the second item that we have to help with getting blood flow back to the heart is contraction of our muscles. So you can see right here, as the muscle is relaxed, blood could flow, no problem. But as we have contraction of these muscles, it will actually squeeze that blood. All right, we won't squeeze it back past that valve. That valve's closed. But from this area, it would have been squeezing the blood upward. All right, so our muscles actually can go through this milking action. All right, so as the muscle contracts, it's going to compress that vein and push the blood forward. All right, and you'll open up the valves, pushing it up and in. As they relax, as that blood may start to backflow, those valves will slam shut and the blood's been moved along. Okay, so like I said, it's this milking process, all right, and only allows blood to flow in one direction. But this is where we can run into problems. If these valves do not work properly, blood will start to pool in those veins, and this causes distension within those veins. All right, and this is where we start to see cosmetic issues as well as some mild discomfort, because we see those as those varicose veins. All right, as it starts to pool in there and backflows, that it'll start to spread out. All right, and that's why we see these varicose veins or spider veins that start to occur. All right, and as it continues to build, we may start to see painful distortion within adjacent tissues. All right, and this may be what you see down at the anus. So hemorrhoids. All right, hemorrhoids are a painful distortion of that venous blood. All right, and it's just a matter of now that vessel starts to push outward. All right, and last thing to talk about within this session is when we look at total blood volume distribution. All right, where do we find all of it? Well, for the most part, it's sitting within our venous system. That our venous system is our storage area. Okay, so it's unevenly distributed between our arteries, veins, and capillaries. All right, with that venous side containing about 64%. Okay, and with it being 64%, if we look at a normal adult having upwards of about 5.5 to 6 liters of blood, that means this is about 3.5 liters sitting in the venous system. All right, about 1 liter of that is within the venous network, carrying blood from our digestive organs back to the liver. So that's a huge area. All right, and then our systemic arteries will make up about 13%, being the next largest item. Okay, and then the remaining amount of blood is within our pulmonary circuit and then also in the heart and systemic capillaries. All right. Another item, since we're on this area, to talk about would be venoconstriction. Okay? And venoconstriction being that concept of when we contract that smooth muscle. All right, so that's tunica media. All right, this will help to reduce that diameter of the veins and also the amount of blood that's within our venous system. Okay, and this is how we maintain blood volume. So especially if we become active, we're going to constrict our venous side. We want more blood flowing through. So rather than being that reservoir system, we need that blood out and moving. Okay, but of course, if we're relaxed, then basically we don't need all that blood. So we can allow it to hang out in the venous system and kind of pool, but remember, it's not pushing back on those veins. It's just a matter of we've gone into our relaxation. All right, and just realize venoconstriction is controlled by our sympathetic nerves. All right, and just realize what I was just discussing here is venoconstriction, whereas when we look at it as a whole, whether it's the arteries or if it's the veins, we can talk about vasoconstriction for the vessels themselves. All right, but if we talk about just the veins as far as how we control our reservoir or supplies, that then gets into venoconstriction. All right, and then the last item that I've been using throughout this is just that term distensibility. That is our ability to stretch and expand. All right, just making sure that we define that clearly. And that now brings us to the end of this session.